Sir, in 5 minutes, sir, we'll start, sir. Fakruddin, sir. Fakruddin, sir. In 5 minutes, we'll start, sir.
Yes, sir, Srinivas.
Yes, sir. In 5 minutes,
Yeah.
we'll start, sir.
Yeah, sure.
Yes. Fakruddin, sir. Sir.
Yes,
Good evening.
Srinivas.
Good evening, sir.
Good evening.
Shall we start, sir?
Can we start?
Yes, sir.
Am
Sir,
I
I will.
audible? My volume is
Yes.
okay?
Yes, sir. Yes, sir.
Yeah.
So, good evening once again to everybody, and I wish you happy Krishna Janmashtami. Today we have Dr. B. Prakruddin, sir. Sir is a professor and head of the Department of Biotechnology and Crop Improvement at College of Horticulture, University of Agricultural Sciences, Bangalore. Professor B. Fakruddin obtained BSc Agriculture, MSc Agriculture, and PhD in Genetics and Plant Breeding from University of Agricultural Sciences, Bangalore, and postdoctoral training from George August University, Göttingen, Germany, and the University of Missouri, United States of America. Sir possesses PG Diploma in Bioinformatics, BIA Noida, PG Diploma in Agriculture Extension Management from Manage Hyderabad, and a certificate course in Genetic Engineering from IISc Bangalore. Sir served at the Department of Biotechnology, University of Agricultural Sciences, Dharwad from 2002 to 2013 and moved to University of Horticultural Sciences, Bagalkot as full professor of Plant Biotechnology in 2013 and since then serving as professor and at the Department of Biotechnology and Crop Improvement. Sir is founder faculty of the Institute of Agri-Biotechnology, IABT, and initiated the Nanotechnology Laboratory at University of Agricultural Sciences, Dharwad, and Center of Biotechnology Research at UHS Bagalkot. Sir has guided 53 masters and 15 doctoral students as a major advisor, besides mentoring 6 postdoctoral students. Sir has trained over 100 faculty scientists through ICR funded summer and CAFT programs in biotechnology and plant genetic resources. Sir has published over 210 research papers in national and international journals, 15 book chapters, 28 reports, and 62 abstracts in conferences and also in seminars. Sir has released 6 varieties at UHS Bagalkot and developed BT brinjal and BT pigeon tea besides multiple QTL introgressed sorghum lines for drought and charcoal rot tolerance at UAS Dharwad and granted with an Indian patent. His laboratory has been well funded both at the University of Agricultural Sciences Dharwad and also University of Horticultural Sciences Bagalkot from different funding agencies such as DRDO, DBT, ICAR, DST, BBRC UK, Indo-US Agriculture Knowledge Initiative, Indo-USA Diploma, uh, USA Obama's Singh 21st Century Knowledge Initiative, ITBT and SNT Government of Karnataka, RKVY Government of Karnataka, etc. Sir was awarded the Sir C.V. Raman Young Scientist Award, Government of Karnataka, Best Research Scientist Award, UHS Bagalkot, Incentive Award for mobilizing maximum product grants at University of Agricultural Sciences, Dharwad. With this brief introduction, once again, on behalf of all of you, KSTA, KIDS, DBT Government of India, and my own behalf, we welcome you, sir, for giving the talk on CRISPR technology. So over to you, sir, please.
Thank you. Thank you, Srinivas, for your generous and kind introduction. Good evening to everybody once again. And my talk, I have planned it for about 45 minutes. And this covers largely what are the big problems that we are facing and how these cutting edge technologies are offering ways and means to overcome these problems. And particularly, we will look at the genome editing, the CRISPR in particular. CRISPR, the clustered, regularly interspaced, short palindromic repeats, is an technology that has potential applications right now, and it is a cutting edge way of engineering the genomes and altering the genetic variations that we need. This is giving a sort of opportunity that is like highly specific and more precise, unlike general genetic engineering approaches that we had hitherto. As I mentioned, to look at what are the problems that we have right now, the big problems to say, like we all know that the increase in demography, the population is really boosting the demand for the food. Why I wanted to say this is something that is very important and basic. Current world population with its number at 7.6 billion or so is likely to become 10 billions by 2050. But as per yet another estimation, which is expected to grow at about 33%, it is also likely to reach about 16.5 billion. That's a lot of population in a short period to come onto the come over to the earth. And then another big thing that is happening is urbanization is increasing. Urbanization is increasing, and this one particular aspect of increasing urbanization, it is estimated that a net addition of about 2.4 billion people to the towns and cities. This trend is in fact rising. This will generally increase in the moving people to the towns and cities will increase the income, and then as income increases, you know that processed foods will be, the consumption of processed foods will be more, and that leads to child obesity. Although this looks like a small thing, but this is a big problem. With this, what's happening is, like, from 10 billion to, we will have to produce about 70% more food, and also the consumption of meat, which is at 36.4 kgs per person annually, is likely to become 45.3 kgs per person annually by 2030. That means a lot of stress on the environment to produce so much of meat. The second big thing that we are concerned right now is current use of natural resources are highly stressed. After population, this is the next big thing which we are concerned about. Natural resources are highly stressed. The world farmland is increasingly becoming unsuitable for production. At present, about 25% of the land is already rated as highly degraded, in fact, all over the world taking. And now about 44% is considered as moderately or slightly degraded. It is just a matter of time that this 44% would be added to this 25%. That means a lot of land is going to be unsuitable for production. So world water resources are highly stressed. In fact, more than 40% of the world rural population is living in a water scarce area. And you all know now, like, you know, we depend, in fact, don't consume the natural water just like that, the water that is coming from tap. We generally go for the filtration and then consume the water. That is the state of the affair, and we know all of us are facing right now that aspect of it. So the water resources are highly stressed right now that way, and it is going to be much more with the increase in the population. Then the land shortage has resulted in smaller farms. This is another aspect that has the cascading effects indeed. This generally results in the lower productivity, and that adds to the poverty. So this is another thing which is a big thing that we need to be worried about. And we all should know that most of these things are the primary cause for this is agriculture. Agriculture, while is a noble profession, and agriculture is noble by itself because it feeds the world, but it also is responsible. This particular agriculture is also responsible. Agriculture and related activities are also responsible for putting a lot of stress on the natural resources. And another aspect of this is, like, direct and indirect causes of land degradation are all those activities which are responsible for direct and indirect causes of land degradation are related to agriculture, be it deforestation, be it overcutting of vegetation, be it inadequate fallow periods, because we are not allowing any fallow periods at all. We are overusing them, overgrazing, improper crop rotation, urban and unbalanced fertilizer usage. All these things, all these activities, if you see, these are all responsible for the land degradation, are all related to agriculture in one or other way. Now, the third big thing that we need to be worried about is climate change. Climate change is really reducing the productivity. This is becoming much more clearer. We had some kind of like doubt about it, but now it is very clear that climate change is a big thing, and it is reducing the productivity, agriculture productivity. Agriculture is one of the primary producers of the greenhouse gases, and climate change will affect the affect every aspect of food production for that matter. And without efforts to adapt to the climate change, food insecurity will increase substantially as we move on. So that is the aspect that we need to be worried about. Now, if you look at this particular cartoon, agriculture contributes almost twice the greenhouse gases than any other related industry by itself, like be it forestry, be it other land uses and everything. It produces twice as much as that, and then net result is reduction in the productivity of the agriculture farms. Now, the fourth and very important aspect that goes unseen, and most of us many a times we contribute to this, is food wastage. Food wastage is a massive market inefficiency, and in fact it is an environmental threat. Food wastage, the food that has been produced with all these difficulties, now if it goes wasted, you just look what is going to happen. So that is the big thing that we are worried about right now. Meanwhile, 800 million people are going to the bed hungry every night. But look at here, about 33 to 50% of the food produced is going waste. It is not consumed at all. About 30 to 50% of the food produced is going waste without uneaten. This is equal to about 25% of the fresh water consumed globally. And if this food wastage were to be a country, it would have been just next to China and US in its contribution to the greenhouse gases. This is the way the cascadal effects they have and the interlinking effects they have eventually onto the environment. So the result of all this thing is outcome is poverty and hunger, and we somehow need to, like, demography is increasing and climate change and food wastage, all these demography, climate change, natural resources and food wastage, all these things are responsible for this. And unless we have some strategies that are going to produce more food, it's very difficult indeed to sustain the productivity of our farms as we move ahead. Now, if you look at it, what are these new advancements that are there, which are highly relevant, which are cutting edge in their nature? If you look at it, plant breeding tools, in fact, the toolkit of plant breeding is getting added with newer and newer tools all the time. Plant breeding, making the plant breeding happen much faster, more predictable, and amenable to a wide range of species. These are the tools that we look for. The advances in plant breeding is responsible for faster breeding, more predictable breeding, and one could use these tools to a wide range of species. In fact, several techniques of plant genetic engineering have been developed so far, and they have been used. But the ones, the terminologies that we have come across are like genome engineering, genome editing, gene editing. These are something that are very recent in nature. Actually, either it is genome engineering, genome editing, gene editing, any of these terminologies, they refer to modifications in the genomes of living organisms. And modifications happen how? They happen in terms of either insertions of some nucleotides, or deletion of some nucleotides, or substitution of some nucleotides. Any three of these in their own way are responsible eventually for a genome engineering or genetic engineering or genome editing or gene editing aspect to happen. Now, this genome editing, if you look at it, genome editing through programmable endonucleases is the most recent approach in the genetic engineering. So we use endonucleases which are programmable. One can program these endonucleases, and that is the recent approach in genetic engineering. These endonucleases are used to specifically induce double-strand breaks in target gene of interest. There are two things here. We can induce the double-strand breaks using these endonucleases, and that too in a target gene. It is not like anywhere in the genome. We can specify the gene and induce these breaks. How does this happen? It happens through non-homologous end joining, NHEJ, non-homologous end joining, or through homology directed repeat, HDR. So these are the two ways by which this particular aspect happens. One is non-homologous end joining. Once these breaks are happening, and then it is repaired through non-homologous end joining or through homology directed repair. Now, these things are like cellular repair pathways, then act upon these double strands and then try to restore this damage, this double strand breaks that has happened. And in the process, what happens is there are going to be addition of a few bases, or deletion of a few bases, or substitution of a few bases, or sometimes recombination of the DNA can happen in this process. So basically, double strand breaks are induced, and these breaks are like this happens through non-homologous end joining or homology directed repair. And then during this process, these insertion, deletion, substitutions, or recombinations can happen in the genome. Now, the principle of these non-homologous end joining or homology directed end joining, and most of you know. So in a non-homologous end joining, there can be a deletion of various lengths. It could be less, more, or various length deletions can happen. Similarly, variable lengths of indels can happen. Either insertions or deletions can happen in the DNA. In a homology directed repair mechanism, you have a donor and you have a template, and there is a homology between these two. When there is a homology, the homology directed change is going to happen, and generally this change, this insertion, deletion, or anything that is going to happen here, this is going to be a very precise insertion or a modification that is going to happen over here. So we have a kind of definition for this. It happens definitely. The variability word is seldom over here. Now, there are three ways in which these things happen. There are three kinds of programmable endonucleases. We're talking about those, the programmable endonucleases that are used in the plant genome editing. Which are those three? These are those three. Zinc finger nucleases, ZFNs, transcription activator like effector nucleases, commonly called as talons. And then the latest tool in this series are the logical evolution of these talons is the CRISPR-Cas system. So these are the three ways in which these endonucleases are used for plant genome editing. Briefly, let us look at each one of these three and maybe a little deeper into the third one. These zinc finger nucleases, these nucleases are chimeric proteins that are composed of a synthetic zinc finger DNA binding domain. So you have a synthetic DNA binding domain, right, where the zinc fingerprint DNA domain, and a DNA cleavage domain. You have a protein component. Now, this is a hybrid restriction enzyme system. You have a DNA and then you have a protein, and that serves as a restriction system. You can see the protein over here, a DNA cleavage domain, and then you have a synthetic zinc finger DNA binding domain over here. So this projects like a triplet. The recognition of DNA is in fact triplet DNA manner, and the zinc finger DNA binding domain can be modified to the specific target of any length stretch of DNA, double-stranded DNA of interest. So that gives the capability for us now to target different lengths of DNA, target DNA. So now they are like they were first identified DNA binding motif from the transcription factor 3A from the African cloud frog, and the small protein structural motif that is characterized by the coordination of one or more zinc ions in order to stabilize this particular unit or this fold is associated with this, and it contains multiple zinc finger like protrusions. That's why is this name, and that make the tandem contact with their target module, just like this. You can see here left and right. So and this structure I showed to you already. So you have, it consists of two parts as I mentioned. There is a FOC1, like FOC1 naturally found in the flower bacteria Vacanocatis, and then the N-terminal binding domain and a non-specific DNA cleavage domain at the C-terminal. Now these, like as shown, adds a donor DNA or a non-homologous end joining manner, variable and much precise here, and as we saw already, this happens with this. So these zinc fingerprint nucleases, in fact, for good period, they have been in use. They have been used in like many crops, like maize or rapeseed. There are a lot of reports. Just to show you, it's like inhibiting the replication of begomoviruses using artificial zinc finger nucleases that target viral conserved nucleotide motif. So begomoviruses is a group of viruses that cause leaf curl disease in tomato, and they targeted a conserved domain in the begomoviruses, and then they designed the artificial zinc finger nuclease to that, and then they used it over there in order to inhibit the replication of the begomoviruses. So you identify the target site like this in the begomovirus, and then design the zinc finger, artificial zinc finger designs like this, and then use them. So you can see here, when they use the artificial zinc finger nucleases, you can see the digested DNA over here. Target DNA was, you can see it here, right? So, and then when they titered the the viruses using QRT-PCR, in fact, wherever there was a treatment of this artificial zinc nucleotides, the titer was low, in the sense the viral replication was inhibited. So that means it works. So there are a large number of publications like this targeting specific diseases and specific aspects of the interest of the scientist. Now the other one is transcription activator like effector nucleotides, talents. Here the talents are sequence specific nucleases and consisting of transcription activator like effector, which is fused to the catalytic domain of the fork1 endonuclease, the same fork1 endonuclease. Now the DNA binding domain in a tail monomer in turn is comprised of a central domain, central repeat domain, and this central repeat domain directs the DNA binding and host specificity, the CDR. The CDR is formed by tandem repeats of 34 amino acid residues, each binding to one nucleotide in target nucleotide sequence. There are 34 amino acid residues in that domain, and each one of them bind to one nucleotide in target nucleotide sequence. This allows more flexible, you have 34 nucleotides, so therefore it allows more flexible target design and increases the number of potential targets that can be targeted in the specific region. So it consists of basically a tail and an endonuclease, and then it binds specific DNA nucleotide, binds with a specific DNA nucleotide, and it was first repeated by Ullabonas in Xanthomonas variza in 1989, and in fact for a long time these were used. Tails are organized into three sections. Like you basically have a N-terminal domain, which consists of type 3 secretion signal, and then you have a central repeat domain that determines the DNA binding specificity, and then you have a C-terminal domain containing a nuclear localization, NLS, basically nuclear localization signal, and an acidic activation domain. Now, the stretch of this 34 amino acids, they can each repeat amino acid at the position 1, 2, this side over here, and 1, 3, this side, left and right over here. These varies. Thus, this repeat variable diresidues, RVDs, this repeat variable diresidues play a critical role here. These repeat variable diresidues are critical. The amino acid identity of the RBDs is responsible for the DNA nucleotide recognition and enabling the design of the talents to target this unique DNA sequence which we are interested in. And then this works this way, as I showed in the previous slide. Now you have basically bases and then the analog amino acids in the tail. These are the amino acids in the tail, and then you have the dictionary to choose from. And we have a good number of ways in which you can design the target tails over here. So these are some of the sites which you can visit and then try to make use of them to design the strategy. Now, using this particular system, a good number of papers have come out, and this has been used heavily in plant system, like targeted mutagenesis of tomato procera gene using transcription activator like effector nucleases. So they chose clearly a thing, and you can now see here the plants that are edited. You can clearly see the plants that are edited. Now coming to the third, such many publications are there to see for. Now let's come to this CRISPR-Cas system. This CRISPR-Cas system, this CRISPR Cas system, it consists of a programmable, again, Cas9 nuclease. There is a Cas9 nuclease which is programmable, and there is a synthetic short guide RNA, sgRNA, short guide RNA. So there is a short guide RNA, and then there is a nuclease enzyme. So the combination of these two. Now here the target DNA target specificity is provided by the guide RNA. So the name indicates it is the guide. It guides to the specific sequence of our interest. So guide RNA provides the specificity for this nucleus to go on docked. So therefore, this CRISPR-Cas system is much more easier compared to either zinc fingers or TALEN system, because here we can program the CRISPR-Cas9 much more easily than either zinc finger or TALEN system. And a few more things related to this is, like, it is simple, efficient, and has a low cost, and allows targeting of multiple genes at once in a genome. So that is also possible. Now this CRISPR-Cas9 system has a wide applications in agriculture, medicine, and in multiple fields. In medicine, for example, we keep reading, you all might have read it as well. It has been applied in research related to cancer, viruses, disease, many pathogens in many domains. It has been applied. So basically, you have a Cas9 protein over here, and you have a the single guide RNA, and so a single guide RNA becomes part of Cas9, and this is the target DNA over here. And now this target guide RNA targets this sequence because it is designed that way. And now the Cas9 works upon and introduces the double strand nicks here. And once these nicks are there, it falls into the already known pathways of homology directed DNA repair, as we saw, or non-homologous end joining pathways which are there already in the cellular system. So this is how this begins. Now these CRISPRs, basically, they are a family of DNA sequences. So these are a family of DNA sequences. Basically, they are found in the genomes of bacteria and archaea, and they were first discovered in the downstream alkaline phosphatase isozyme gene, IAP, like 1987 by Ishino. I'll try to show you this abstract. And then these palindromic repeats are separated by about 32 to 36 sequences derived from DNA of viruses that previously infected the bacteria. So that means these repeats are separated by the sequences that are derived from the viruses that infected the bacteria previously. So that means the moment the bacteria infects the virus infects the bacteria, a few sequences there become part of the bacteria, right? And they remain between these repeats, these palindromic repeats, and that's how they act as the memory. And then these viral derived sequences integrated into the bacterial genome provide as a memory system of the virus that infected. And as the virus comes, once they integrate into genome, the CRISPR system activates, and then Cas9 associated protein will be able to degrade that once that virus comes in. Just like this, see, you look at this red, this virus infected, and this becomes part of this cluster or the CRISPR locus. And now this entire CRISPR locus with multiple spacers like this, each spacer having derived from a different virus which infected previously, now gets transcribed, and all these are available as the pre-cRNAs, and they become the part with the Cas9. And whichever the virus, if it infected previously, because the memory is there, that will be targeted through this cRNA, and then it is destroyed. So that's how this immune system works. Now these Cas9 is a as I mentioned to you already, Cas9 is a DNA endonuclease, and it is found in bacteria and archaea. The natural role of CRISPR-Cas9 is basically, as I mentioned to you already, antiviral immunity against the DNA viruses. Now this binary complex system, formed by the guide RNA, sgRNA, as we were talking, and this endonuclease Cas9 recognizes, cleaves the DNA of incoming viruses with this in a very sequence specific manner. And there are several CRISPR-Cas systems, in fact, in bacteria and archaea. And CRISPR-Cas9 system most frequently used in plant genome editing is it's kind of adaptation of the type 2 CRISPR-Cas system from the Streptococcus. Right now, this type 2 CRISPR-Cas system, it consists of a Cas9 protein with DNA endonuclease activity, the Cas9 protein which has the endonuclease activity, and one CRISPR RNA transcript, that is processed from one or several guide RNAs. Okay, and this whole thing directs these guide RNAs that direct Cas9 to target the sequence. So we basically have a guide RNA and then the this endonuclease together to function. I was mentioning to you on this Ishino. In fact, for the first time, the hint towards this CRISPR came from Ishino and his team in like 20 years ago, more than 20 years ago. It was like 20 years ago, 2002, where he gave this name: clustered, regularly interspaced, short palindromic repeats. In fact, at that time, in his paper, nucleotide sequence of the IAP gene responsible for alkaline phosphatase association conversion in the Escherichia coli, identification of a gene product from Japan. See, you can see it clearly here. He talks about it. An unusual structure was found in 3' flanking region of IAP gene. 5 highly homologous sequences of 29 nucleotides were arranged as a direct repeats with 32 nucleotides as spacer. We have been talking about the spacers, right? And then the first sequence was included. I mean, he keeps writing and very clearly says here that so far no sequence homology to these sequences have been found elsewhere in the prokaryotes, and biological significance of this remains unknown. So this we are talking at that time. So, and later the same thing was discovered basically. In 2020, exactly about 20 years later, the Nobel Prize for in Chemistry was awarded to Emmanuel Charpentier and then the Jennifer Doudna, one from the Germany, and Doudna from UC Berkeley. Both of them were awarded in the chemistry. The Nobel Prize was awarded in chemistry for their revolutionary discovery. This became a significant event. Now let us look into the components of this CRISPR and Cas9. So if you look into this CRISPR, basically, like we have 2 to 6 base DNA sequence adjacent to a target DNA, 2 to 6 base sequence adjacent to a target DNA, which is required for effective target recognition. So you have this PAM. It is a protospacer adjacent motif called PAM. These are basically 2 to 6 DNA sequences just adjacent to the target sequence. It is required for effective recognition of the site. Then the second component is CRISPR RNA or CRRNA. This is complementary to target sites, very critical one, complementary to target sites. It is CRRNA. Then we have trans-activating CRRNA, trans-CRRNA. These trans-encoded small RNAs with 24 nucleotide complementary to repeat region of the CRRNA product, I mean precursor transcript. So if you look at it, this is how it is. You see this trans-CRRNA over here, and then the PAM that we are talking 2 to 6 nucleotides over here, and then you have target specific CRRNA over here, and in the background you can clearly see the Cas9 working on the target DNA like this, and this is the position where the double-stranded nicking is going to happen by this Cas9 in a highly specific manner, because we have this target specificity guided to this particular position by this target specific CRRNA. And now this protospacers are like this, which are going to be transcribed, and then you have tracer cRNA, trans cRNA molecules like this, and then this trans cRNA and then the cRNA, the complementary pairing happens like this, and now they become part of this Cas9, and then this region is going to identify the target like this, and then Cas9 cleaves that region. If you look at little details into the Cas9 protein, it's basically a two signature domain protein. It has basically two domains, HNH domain and RUVC domain. This HNH domain, it's like a nucleus domain. It cleaves the target DNA basically. This is the domain that cleaves the target DNA, complemented to the guide RNA, of course. And then this another domain, the RUVC, it's also a nucleus domain. It cleaves non-target strand. So this HNH cleaves the target strand. This cleaves the non-targeted strand. Now this creates a double-stranded break, DSB, right, at the target site, which can be subsequently used to introduce, which gets modified or which gets repaired through non-homologous or homology-directed repair mechanism of the cellular system. In higher plants, it has been shown that NHE, non-homologous end rejoining, occurs more frequently than the homologous directed repair. This requires, so that means because this requires a donor template during the homologous recombination, this one. So double strand breaks during the double strand breaks. This homologous non-end joining does not require a homologous homologous repair template as such. So therefore, this non-homologous end joining has become more popular because you can go multiple places and multiple targets one can try to have, and disrupt the gene by creating some small indels basically. So just to create indels, this non-homologous, this is more popular. As we discussed already, HDR is more precise, while HE, this non-homologous end joining, is not very precise. The additional indels are variable. So, but that is good enough to create the disruption in the gene. So this is how this works. If you try to look very little more details into the CRISPR-Cas thing here, this is the non-targeted DNA. This is the targeted strand. This is the non-targeted strand. On targeted strand, the nucleus domain over here, and this is the on the other side, and this is where is the PAM sequences that we are talking about. And of course, you have the guide RNA over here, which basically directs this Cas9 to this double-stranded region, this specific region. We were also talking about the specific sequence that targets this region, that is the target strand and non-target strand from Cas9. You can also see it here much more, a little more illustratively. So you have HNH over here, which targets the target strand. You also can see the targeted, this particular strand is targeted through this homology binding, and then this is the non-target strand, and you have RUVC domain, and this is exactly the position where the nicking is going to happen. And just addition to that, you have 2 to 6 nucleotides that are critical we were talking about. These are those sequences. So the same thing shown here. Now there are so this is the basic system basically. Now the genome editing vectors are like you have basically a vector component. So you have the guide RNA target sequence, you have scaffold over here, and then you have the promoter system, because you need to transcribe this. You have the promoter system, then the end, the termination system. Then you have other promoter systems as well for the Cas9. So for both you have this is the basic vector that is going to be there, and this vector can be transferred into we can use this vector and a transformation system like Agrobacterium tumefaciens or maybe Rhizogens. A transformation system can be used. Once you have the plant transformed, and then one can use any of these methods that are in vogue, like embryos or ovules, flowers, protoplast, root cell, leaf, or any of these things. It can, the tDNA is going to integrate. Anyway, it is going to integrate randomly. The tDNA integration is random. The expression of Cas9 protein and guide RNA lead to editing of the target DNA. See, once the although it is in tDNA insertion, which is random, once this transcription happens, the editing is going to be highly precise. It's going to be a target editing. Targeted DNA only is going to be edited. The DNA, tDNA insert and insertion site, and then DNA targets are likely not linked to each other anyway. Then with the DNA, tDNA insertion and edited plants of genome, they can be like separated through the Mendelian segregation in the subsequent generations. So there are a good number of sites, softwares which allows one to design these gRNAs, TALENs, NFs, as I also showed in the previously couple of slides. For CRISPR design tools are available at this MIT site. It helps to design gRNA sequences that more with less off targets. There are multiple sites like this that one can try to make use of them. So as I showed in previously, like there are a large number of papers, in fact, efficient CRISPR-Cas based gene modification system for the watermelon, for example, this paper. So very clearly they target the sequence, and then they use this and very clearly show that this watermelon, the gene is disrupted. Cas9 mediated DMR6 ortholog of tomato was edited, and then they showed that it confers broad spectrum disease resistance. See, you can see the wild type and then DMR6 mutant with less. So one can take there are many examples, like you can look at Arabidopsis thaliana, rice, a good number of, in fact, a large number of many crop plants have been used. In many crop plants, this tool has been used. There are this tool, as you also might have read and heard, like there are many licensing type of things that have happened. The large number of licenses that have gone in, both exclusive and non-exclusive, by the IP owners. Both exclusive and non-exclusive licensing has happened into this. It's only the beginning. A large versions of this CRISPR is likely to come as we go on. If you look at the application of this gene editing technologies, you can see human therapeutics, the number of companies that are using, like about 15 companies. Biotech research, a good number of companies. If you look at like blood disorders, cancer, if you look at different, these are the sectors, and these are different diseases, HIV, cancer, genetic, many diseases. People are using this gene editing technology to address the issue there. This is a wonderful tool. In fact, the future of CRISPR-Cas9 genome editing is likely to be like definitely into the agriculture. The good is India actually deregulated this, permitted specific types of gene editing products just about 2 to 3 months ago. 3 months ago, the verdict came. I mean, unlike general genetic engineering products, the edited products are now permitted have been allowed in our country, deregulated in our country. That's a very good beginning, and very likely that these products are getting into market very soon. So it is used for IP, the drug screening, human gene therapy, agriculture. For agriculture includes animals, crops, and horticulture, many things. This tool is wonderfully useful in synthetic biology, pathway engineering, and then programmable RNA targeting, viral gene disruptions, pathogen gene disruption. In fact, and then also like ecological vector control, mosquito sterilization, many applications. Applications are plenty. I work with tomato, and I generally keep in a large number of publications, and a large number of genes have already been edited and reported in tomato. Same is the case in multiple crops. So the only concern here is like of the version that is available. Multiple versions of CRISPRs are available. Of the versions that are now in vogue to use, it requires prior sequence information. So when you want to design a guide RNA, we need to know what is the gene sequence that we are targeting. So that is one requirement as of now. And then another concern is off-target mutations. But off-target mutations, there are also ways in which one can try to manage all these, as it is an evolving tool. It is just beginning, and there are going to be many more versions of that that are likely to come, and many of these concerns can be addressed with. And then with respect to prior genome sequence information, as you know, the sequencing is becoming much more possible now, much more easier now, and much more affordable now. And it's very likely that we will have sequence information for most of our croplands. In fact, draft sequences of many of our croplands are available. Model plants, model crops, and model plant, they have been well sequenced, and a deep sequence information is available anyway for the basic research. But for the application, like farm animals and farm crops, there are good number of major crops that have been sequenced, and a very good sequence genome sequence is available. But it is also very likely that even minor crops are also going to be sequenced as we move ahead, because the sequence cost is coming down, accuracy is increasing, the tools, the genome sequencing platforms are becoming more affordable and more efficient and good. So this is what I had to share with you. Thank you very much, and if you have any questions, I'll be happy to take them.
Thank you, sir. Any other questions from the participants? Any questions?
As of now, there are no questions in the chat box and the Q&A section. If there are any questions from the audience, please raise your virtual hand so that we can unmute you. You can discuss with them directly, and you can get clarified all your queries. If there are no questions, we can go to the next session.
You also can pass on the questions to me. I'll be able to.
Yeah, sure, sir. Definitely, sir. We
Yes,
will
sir, yes, sir,
write to you, sir.
Yeah.
As of now, there are no questions, sir. If in future they ask any queries,
Sure.
any doubts,
I'll be happy.
we'll
I'll be
update
happy.
you.
Yeah.
Sure, sir. Thank you very much, sir. Thank you.
Thank you, Dr.
Thank
Srinivas.
you very much, sir.
Thank you very much.
Thank you so much,
Thank
sir.
you.
Thank you.
Thank you. Bye.
Bye, sir. Sir, good evening, sir. Suabrna sir. Good evening, sir.
Good evening. Can you hear me?
Yes, sir. Please, sir.
Okay.
Sir.
Okay.
Yes, sir. So, shall we start, sir?
Yeah.
So, welcome back, and we have another scientist director, Dr. Suabrna Rai, sir. Presently, Suabrna Rai, sir, is a director, ICMR National Medicine, Department of Health Research, Government of India. It is Belgavi. Sir did BSc, MSc, PhD from Sir, hi, sir. Diseases, laboratories at the ICMR National Medical Research Center. At Belgavi in North Karnataka in 2009, and collaborated with the state health services and various government medical colleges to identify, investigate, and control outbreaks of infectious diseases like cholera, diarrhea, and diphtheria, hepatitis, etc. This laboratory later led the way in COVID-19 testing in North Karnataka, and trained medical college staff from across the region since early 2020 when the pandemic started. Dr. Rai sir is also a member of the Karnataka State COVID-19 Task Force for genomic surveillance of SARS-CoV-2, played a major role in estimating the burden of rotavirus diarrhea in children arts of North Karnataka and southwestern Maharashtra, and identified the circulating strains in the ICMR nationwide network project that led to introduction of rotavirus vaccination in the Universal Immunization Program. Dr. Subarna Rai sir was also the first to flag resurgence of diphtheria in Karnataka state, highlighting the emergence of the disease and increase in deaths. He was involved in advocacy of availability of DAT in affected areas to reduce deaths through policy briefs sent to Government of Karnataka through ICMR New Delhi. Sir has also conducted national sero survey for diphtheria antibodies in various groups of children and flagged waning immunity in older children, calling for better DPT coverage and appropriate booster dosage. Professor Roy sir worked on lives with the Government of Karnataka, Department of Health and Family Welfare, and Department of Medical Education for establishment of model rural health research at Sirva, Raichur district, which is working on improvement of health status of deprived and marginalized people of the region. Served as nodal officer of the OHR scheme since inception. Sir is also routinely provides training, technical and laboratory support to the state government, the Department of Health and Family Welfare Services. And also, sir has contributed that 7 book chapters, policy briefs to identify a potential relate for food using reverse has been prestigious Mark Sahib Singh Soke Award, the highest content of Nicobar of tsunami. 2004, Misra of infertility, leptospires. So with this brief introduction, so thank you so much, sir, for accepting our invitation to give a talk on big data biology in health and research on behalf of all of you and Karnataka Innovation and Technology Society, Karnataka Science and Technology Academy, and DBT Government of India on behalf of all the attendees. Warm welcome to you, sir, for giving this talk, sir.
Thank you. Thank you so much.
Yes, sir.
So I will share my slide.
Sir, please.
Is it visible?
Yes,
Yes, sir.
yes, sir. Yes, sir.
Okay. Thank you. First of all, let me express my sincere gratitude and thanks to KSTA for inviting me and giving me this opportunity to speak to the audience, and also the DBT for organizing such a wonderful program. And yeah, so what I will do is that I will start with my presentation. So if there is any disturbance in between, please stop me so that I can reorganize. There might be some audio or visual problems, or if the slides are not moving, please let me know.
Sure, sir. Thank you, sir. Please go ahead.
Yeah.
Yes, sir.
Okay. So basically, I will be speaking on this big data biology in health and research. And is the slide moving? I hope that the slide is moving, isn't it? Am I audible?
Yes, sir. Audible, sir.
Okay, yeah. So what is big data? Big data are actually data sets that are too large or sometimes more complicated to be dealt with by the traditional data processing application softwares. Big data challenges include the sampling and capturing of data, data storage, data analytics, search, sharing, transfer, visualization, querying, updating, analysis, information privacy, and data source. So there are 6 V's of big data. The first 3 are very important, which started with is the volume, variety, and velocity. Volume is the amount of data from a myriad of sources. It can be from anywhere. Variety is, as I said, it can be from anywhere. So the variety of the data also can be of various types and in various forms, like structured, semi-structured, and unstructured. And velocity is the pace at which you generate that data. Sometimes the data can come fast, sometimes it can come slow. With passage of time, 3 more V's are added to it. First is the veracity. So that means the degree to which you can trust that data, because that is extremely important, because these days we have so much of data, but how much this data is trustworthy is often a question. Then the value. What is the value, the business value of that data collected? How you are going to use it? What will be its utility? And then the variability. So the way in which the data is collected, used, and formatted also will change with your applications. So these are kind of things that we keep in mind while dealing with big data. Examples are Aadhaar, the UIDI is a classic example of a huge amount of data that we have for all across our countrymen. The income tax, the IT data that is with the government on the tax, and that is how they actually even understand tax evasions and all. Sometimes nowadays AI has taken over. So it is an automatic faceless kind of assessment of the taxes that the government collects. The banking data that we all have. So all our banks are linked to our Aadhaar and our PAN card, and a lot of personal details that we have, our expenditure patterns. So these are all linked with our banking systems. More examples, say for example, the ICMR itself maintains a big data of the COVID testing. So from the day one when the testing started till date, and it is updated every day. So this is again a very big data that we have. The COVID portal, it not only has the information about the cases, but also the vaccines. Now we have achieved 200 crore vaccinations. All the details of this are available in this COVID portal. So these are examples of big data. We have email databases, we have our railway and flight, the airline passenger lists, and various numerous databases where we are all involved and our name and details are provided. So, for example, the mobile numbers and the databases that people generally take from various sources, and sometimes we hear that the data is sold in the dark web, and it seems key that there are some hackers who would actually sell this data for marketing purposes and sometimes for fraud also. So these are kind of data that we are talking about. And as you all know now, artificial intelligence is also playing a very important role with this data, because artificial intelligence is also a part of machine learning, where the more authentic and the more data is fed, the better the artificial intelligence becomes. And nowadays, with almost everywhere, artificial intelligence has made its way into. And therefore, the role of big data in artificial intelligence and therefore in our daily lives is extremely important. So now about the informatics boom, this big data biology that we are talking about. So this has been in the last decade or so, the volume of information or the data has been tremendous. If we look at the biological databases, the growth, then we will see that from the late 1990s, there has been an exponential rise in biological databases across the world. If we look at the growth of databases again, if we look at the published genomes of humans or other organisms, you will see the red line here is that it has taken an exponential turn. So we are actually getting huge number of genomic data from all kinds of species together. Data is also collected now faster and in greater volume. remember the another two V's that we talked about, the velocity and the volume. So now both have increased significantly. And as you can see in this particular graph, the validated data is on green, the red is the clustered data, and the blue is the submitted data. So the submitted data is becoming so high that actually validated data is kind of lagging behind. So I talked about the veracity or the dependability of the data that is generated. Many of the data that we generate does not keep pace with, the validation does not keep pace with the generation of the data itself. Medline is the database of medical papers, the medical sciences publications that happen. And there also the number of articles published every year has been increasing again in an exponential scale. Now, if we look at publications, if we look at disciplines, then we will see that the maximum number of publications have been made in multidisciplinary areas, followed by dentistry, veterinary, pharmacology, toxicology and pharmaceutics, biochemistry, genetics and molecular biology, medicine, agricultural and biological sciences, health professions, and then comes physics, astronomy, neuroscience, immunology, microbiology, environmental science, mathematics, social sciences. So if you look at this pattern, then you will realize that the top few are actually from the biology or the biological fields, particularly with the health sciences. Therefore, the data that is generated in the biological field and the health sciences are much more than actually the data that is generated in other disciplines. The same thing happens with the number of publications that we have been seeing. So if you look at this particular graph, here we will see that the red worm is actually the one on bioinformatics. So information science, information technology has come or merged with biology, and bioinformatics is providing a lot of data in which I will be dealing a little bit shortly in the next few slides. Then it is followed by genomics, and then proteomics, and then computational proteomics. There are a few challenges though. Doubling time of the next gen whole genome sequencing data is about 6 months. So today, if we have 10,000 sequences, we expect that the data of 20,000 sequences will happen in the next 6 months. So that means the amount of data we have today doubles in 6 months. However, if we are to look for data storage, then we will see that the doubling time is around 18 months. That means at the same cost, say around for 500 rupees today, if you are buying a 4 GB hard disk or a chip, then at the same cost, you will be able to buy a 8 GB hard disk or a chip after 18 months. So that is the time of doubling time of storage capability. So therefore, if the same rate of costs or the generation continues, then the storing of data, the cost of storing the data will actually exceed the cost of generation of the data in the next few years. The biggest challenge for scientists, therefore, is to make sense of all this enormous amount of data that is made available to us and to utilize them in the best way, optimally, for the benefit of humanity. So this is the PubMed website. As you all know, that is the gold mine for us researchers for all scientific articles published in medical sciences. So that itself is a big database. And if you look at human genome databases, maps, and viewers, you will find quite a number of web resources which actually house these databases of the human genome. Now, a lot of human genomes are being sequenced, and an advantage of that is that now we are able to kind of map various diseases linked to various kinds of mutations in the human genome itself. That has led to the advent of the genomic medicine. The Human Genome Project itself got completed in 2003 and has provided access to the entire human genome sequence for the entire globe. In effect, the genome became a tool for enabling physicians to understand the biological and the disease variability of their patients. So these genetic tests are very powerful because they are, with this we are able to diagnose the disease more precisely. It allows for selection of a specific medication to best treat the disease, predict the risk of disease before the symptoms appear, and manage the disease more effectively. For example, as I said, to diagnose a disease more precisely, if I may say that the acute lymphoblastic leukemia is the most common form of childhood leukemia. So these genetic tests identify these subtypes and types and allow precise treatment and timing for this kind of ailments. And this has resulted in a cure rate of around 80% versus only 4% which was there in the 1960s. Now, for selecting a specific treatment, I can give the example of the Philadelphia chromosome, where a translocation between chromosomes 9 and 22 results in a fusion gene formed from VCR and ABL1 genes. This fusion on this chromosome 22 is called the Philadelphia chromosome and is characterized by chronic myeloid leukemia. This fusion results in increased cellular signaling, causing white blood cell to proliferate. There is a drug called imatinib, which is an oral medication that particularly targets this translocation and it arrests this disorder. Another example of predicting the risk of disease before the symptoms appear. So many of you may be knowing that there are genetic tests which have identified two mutations, one in BRCA1 and the other in BRCA2 genes, that increase the risk for having breast cancer and ovarian cancer in women. And these genetic tests identify greatly the increased hereditary risk for having this breast and ovarian cancer. And knowledge of this increased risk allows preventive measures such as close monitoring, risk avoidance, and even preventive surgery or chemotherapy. So, for example, it has been said that a woman with mutations in BRCA1 and 2 has a risk of developing breast cancer to up to 85%, whereas those without that mutation has only about 13%. Similarly, the lifetime risk of developing ovarian cancer becomes about 45% with the BRCA1 and 2 mutations, and without it, it is only 1.7%. So this knowledge actually led Angelina Jolie to have a vasectomy, and that became a big news and a lot of controversy actually. To what extent we should be taking preemptive steps, particularly for this condition. So, but then these are the news that actually led people to take decisions. Another interesting case I would like to present before you, a screenshot taken yesterday of a particular company called 23andMe. So it says that it says welcome in about $100 you will get a spittoon couriered to you in a kit, and what you have spit on that spittoon, and this tube will collect spit and seal it, and you mail it back to the address given. There is a self-contained mailer, so you mail it back to them, and then after a few days you can log into the 23andMe site, and it will welcome you to yourself, and then you will get to see all the results of the genetic analysis that have been done on your sputum sample, and you will get your clinical reports.
Sir, your voice is not coming, sir. In between, it is interrupting.
Is it?
Yes, sir. Yes, sir. Just now. Yes, sir.
Just a minute. Is it clear now? Is it
Yes,
better?
sir. Yes, sir. Now it is clear, sir.
Okay.
Just a fraction of seconds it was breaking, sir.
Okay. Thank you.
Thank you.
So, so this
Sir, sorry, sir. Again it is breaking, sir.
okay, just
Yes, sir. Now it is coming, sir.
I speak a little slowly.
Maybe some network issue, I think, sir. Yes.
Am I clear now?
Yes, sir. Yes, sir. We can hear you, sir.
So after the research reports,
Sir, again it is breaking, sir. Sorry, sir. Sorry to interrupt, sir. I think you are not using mouse, right, sir?
I'm using actually two microphones. So one
Okay.
trying to change the audio.
Till now it was coming very well, sir. Now.
Okay. So this is about the clinical reviews. And you know the traits. What are the traits that you have? The traits of sickle cell anemia, or the traits of some resistance, or your eye color, or your earwax size. All these details will come. Then you will get. You will also come. So this leads to can have medications for yourself. And this also is part of which is more and this is appreciated systems.
Sir, sorry, sir. Sorry to interrupt, sir. Again.
Just a minute, hold on.
The topic is such an interesting and your slides are very wonderful, sir.
Yeah.
And if we miss the voice, it will be very loss for us also. And it will take time, sir. Yes, sir.
Please hold on.
Yes, sir. Yes, sir.
Is it better now?
Yes, sir. Yes, sir.
Can you hear me now?
Yes, sir. Very well, sir.
Okay. I switched to a different source.
Okay, sir. Oh, thank you, sir.
Yeah. So what I was telling is that in
Sir, from
what we are doing,
from previous
Yeah.
slide you can go, sir. 30-second slide you can start, sir.
30 seconds.
Yes, sir.
Okay. Yeah. So basically, what I was telling is that today's genomic medicine actually gives us all the details about the person, their genetic makeup. You can understand what are the type of risks that we have and what are the type of diseases that can be actually addressed at the genomic level. But then again, here also we have tremendous amount of data, but we are yet to kind of relate which mutations are actually responsible for what kind of disease or what kind of predisposition. As well as there are multifactorial diseases in which multiple genes and multiple factors like even the epigenetic factors will come into picture. So those are areas which leave a lot of scope for us researchers to exploit.
Thank you, sir. Sir, full screen. Can you make it full screen?
Yeah.
Yes. Thank you, sir. Sorry for the interruption, sir.
Yes. No. I'm thankful to you because you are pointing out, so I am trying to correct it. Okay. So what I was telling is that this brings me to the Indian traditional systems of medicine, which has been working on these individualistic approaches, the personalized medicine from times immemorial. And it was also taking into consideration the epigenetic factors like the environment as well as the lifestyles. And we at our institute at NITM is trying to merge this knowledge of traditional medicine with the big data biology so that we are able to validate the use of certain traditional systems of medicine in our country, as well as we are able to bring new drugs and new ways to manage different kinds of diseases that are now emerging. So what we do is that we take this big data for identifying the particular disease or the metabolic pathway that we want to address. Say, for example, if we are working on diabetes, we are trying to understand the various players in glucose metabolism, the kind of pathways that gets altered, the enzymes involved, and then we look at the traditional knowledge. In traditional systems, what are those drugs, what are those herbs that were used for treatment of these diseases? And then what we do is that once we identify one or two plants, then we try to mine the data, again the databases, to understand what are the phytocompounds that have been reported from this particular plants. And then we use the systems biology approach, the computer simulations and high-end computing using bioinformatics to have this disease and the target and drug combinations. So the ones which work most in silico in computer simulations, we take into the validations for the wet lab studies and the animal studies. So that actually reduces time for development of drugs from traditional medicines, as well as it helps us in validating the use of various traditional medicines in various kinds of ailments. And as you all know, that most of the present day drugs have been actually originated from the traditional medicines or from medicinal plants. I have listed here a number of them, but there are innumerable number of plants that are being used, and we have got valuable drugs and medicines from these particular plants. So what we are essentially doing is following the reverse pharmacology approach, where instead of a known target and unknown safety efficacy, we generally take known plants or known drugs with safety and efficacy for an unknown target. So here we have already clinical evidence which is already in practice. Say in Ayurveda or in any other systems of medicine, a certain plant is used for a particular disease. We take that and then we do the preclinical research in vitro, in silico, in animals, in laboratory animals. We do the safety, toxicity, and then efficacy, and then we again carry out limited clinical evaluations in humans. So that is how the reverse pharmacology works, and that is the step that we are following. And for that, again, the bioinformatics and the big data biology plays a very important role. For any strategy for efficient drug discovery, the targets are to reduce cost, and that is done by simulation in computers. To increase data, to increase the velocity of data I talked about in big data, which is multiplexing and doing high throughput. It can be sequencing, it can be anything. To save time, we do parallelization. That means we take many things together. And to reduce errors, human errors, we automate. AI is now replacing that. Therefore, one must be able to generate and cope with a large amount of data in a very short span of time. But most importantly, they must have complete trust on the system and the process that has generated the data. There are several bioinformatic tools for computer simulation. So some of the uses of bioinformatics in drug discovery are in identification of the target disease, which disease you want to target. For screening of interesting compounds, as I said, that we screen quite a number of phytochemical compounds from various traditional treatments that are being given for that particular disease. Detection of molecular basis of disease in question, that means mechanism of action. Rationalization of the use of the drug design techniques. For refinement of compounds based on efficacy and adverse or side effects. And quantitations of structure activity relationship, the QSAR studies. Determination of physical properties, that is solubility of molecules, etc., etc., and the testing of drugs. I'm not going into all the details of this, but a few hints I will give on identification of target disease and interesting compounds. For this, as I said, first the disease which you need to target is identified. Say, for example, diabetes. All that is known about that disease is identified and the structures of key players are determined. So the sugar metabolism is identified as the major pathways in which actually the disease affects us. And then the key players, the enzymes, the pathways, the coenzymes, these are determined. Then their structures are identified. Again, for this structure, you have various data resources in which you can download the structures if they are known. Known existing molecules from hypothesized remedies are identified and their structures are determined. It is also important to look at similar afflictions of their known treatments. So sometimes some of the known treatments will have similar afflictions. So that also has to be identified. Then bioinformatics methods like docking is used to virtually screen these drug target combinations. And it is done like in a lock and key model, whichever fits to the target, whichever likely drug fits into the target, they are taken. You have to understand there can be multiple locks and keys issues. So there can be multiple locks, one key and one key, multiple locks. So you have to be careful about that. The drug must influence the target protein in such a way that it does not interfere with the normal metabolism of the patient. So therefore, you have to be very selective in the action of that particular drug-like candidate that we are screening. Most of the time, compounds that are likely to have some activity against the disease are screened, and they may be only marginally useful. But at some time, there is a way in which chemical modifications can be made to this particular drugs to give it a better drug effect and reduce side effects. So this is what I said here, that drugs can be even tailor-made to suit or to bind at a particular site of the target. And this also can be done computationally as well as in wet lab studies. So it is very important that the determination of what compounds would be tested to be properly determined, that generally depends on the researcher's understanding of the molecular interactions or simply by brute force method. That means you just have a large number of likely drugs which you test against a particular target, and then whichever hit comes, you take it. So that is the brute force method. Otherwise, you are using the researcher's understanding of the molecular interaction and selecting the compounds. Defining of compounds, I'm not going into detail of it, but as I said, that once you identify a loose drug, you can actually make it more tighter as well as much more effective without much of side effects. So you can do that using computer-based technologies as well as in vitro methods. it is very important to identify the 3D structures of the proteins that we are actually looking for, or the likely drugs that we are looking for. Sometimes these 3D structures will not be available. So in that case, what we do is called homology modeling. Here, we generally look at the structures of those compounds which closely resemble our compound in question, but is not exactly that compound. And then the computers actually do, again with artificial intelligence, they will actually model the protein that you are interested in, your actual protein. So even if the actual protein structure is not known, there are ways to predict the structure, the 3D structure, by using computational biology. So one of the softwares that is used is called Modeler, and Swiss model is a repository or a database of such protein structures which are created with this homology modeling. So this is the screenshot of the Modeler software, and this is the right side is the screenshot of the Swiss model where these structures are databased. We use this Autodoc, Schrodinger, and many other tools to actually do this kind of studies. We also have another approach called the subtractive genomic approach. Here, what we often do to find out new or putative drug in drug discovery, here the proteome consisting of all the known proteins of a particular pathogen is retrieved from the database. Now, paralogs or duplicates from that list is removed. That means which are very similar, they are removed with the help of, again, resources like CD hit web server at a certain percentage threshold, which results in a subset of protein sequences. Then we do a BLAST P, and it is done against the human genes, human proteome, to identify non-homologous proteins that yield even fewer non-homologous protein sequences. Now here this is done to see that some of the proteins that may be common in the pathogen as well as in the host, they are removed, because we don't want to target any of the human proteins which are normal to them. So therefore those are removed, and then whichever remains are regarded as those proteins that are essential to that particular pathogen in question. So these proteins we take as targets. So these become very good drug targets, and then we do docking studies from, say, the phytocompounds or modern drugs or repurposed drugs, which was being used very often for developing of drugs against this coronavirus SARS-CoV-2. You have seen in papers that a lot of old drugs have been used in trying to get a treatment for the COVID-19. So this is how this actually were done. So then there are several databases of natural products and herbs related and their chemical constituents. For example, for bioactives, we have the UNPD database, Dr. Duke's phytochemical and ethnobotanical database. We have for chemistry, we have again UNPD, we have ChemBial, we have Drug Bank. For targets, we have BindingDB, ChemBial. We also have SwissDoc. We have PDB, protein database. We have UniProt. For disease target pairs, the associations, we have the TTD, the therapeutic targets database. We have the human protein atlas. For network development and systems biology approach, we have the KEGG pathway, the David, and String as the software tools. For traditional medicine, particularly for the botanicals, the Chinese are have a tremendous amount of data, and that is available in the TCM database, the Chinese traditional medicine database, and you can search them on their chemical names, their TCM name, molecular properties, molecular structure, basic structure, and even advanced search. The herbal ingredients target database is also there, which is a comprehensive and fully curated database of research of protein targets for FDA-approved drugs, as well as promising precursors. It contains about 1,300 known protein targets, and is derived from more than 3,250 literatures, which also covers a lot of Chinese traditional medicines. The UNPD database is a comprehensive resource tool for virtual screening. It comprises of more than 2.5 lakh molecules, and it allows template-based search for molecule and natural source. Supernatural 2 is also a similar database which contains more than 3.25 lakhs of compounds, and their 2D structures, their physicochemical properties, their predicted toxicity, and even potential vendors from whom you can buy these chemicals. There are other resources as well. For example, this Nube resource is from Brazil, which mostly has the Brazilian biodiversity database there. We have Natural Product Alert. TKDL is an Indian one, which it was made, most of you are aware, it was made to prevent piracy of our traditional wisdom and traditional medicinal plants, and to prevent patenting of those by foreign powers. And that lists quite a number of plants and their resources which are used in medicine in India, as well as in other aspects of life. Ayursoft is more of a Ayurveda practitioners database, where a lot of the Ayurveda practices are actually documented in that database. And I will just end with a few of the work that we did. For example, using these techniques. For example, all of you are aware that those people who are under cancer chemotherapy for soft tissue cancers are generally given doxorubicin as an anti-cancer drug. And because of doxorubicin, it is actually extremely toxic to the heart. So therefore, a lot of dose is actually monitored, and then it cannot be given continuously. So it is given in bursts so that the toxicity is not there. And even when we do it in animals, we find quite a lot of toxicity, and with much of problems or much of histopathological differences that occur between normal and doxorubicin treated animals. So what we did is that from a traditional practice, we took cocoa, the cocoa extracts of normal chocolates that are made of. And once they are administered along with doxorubicin, we find that most of the toxicities are removed or reduced in the tested animals. And it is not only with the heart. We have found that even the kidneys and the liver is also protected by the use of concomitant use of coca when somebody is there is treated chemotherapy like doxorubicin. So cocoa can actually help improve the quality of life of people undergoing cancer chemotherapy. So this has been published very recently in PLOS ONE and in Antioxidants. And even for patients on who are suffering from viral hepatitis, we have used similar techniques to relate silymarin for hepatoprotective effect. Similarly, we have found Terminalia cebula to be effective against hepatitis C virus. Against SARS-CoV-2 also, we have quite a number of potential traditional medicine components which are in various stages of testing. None of them have been carried out on humans so far, but at least in the in vitro models, they have shown quite good effectiveness. One for the aging population, we know dementia and Alzheimer's disease is a major problem with the elderly. And what we found is that good old, that red sag, the lal sag, the Amaranthus tricolor, actually is able to reduce the onset of Alzheimer's disease, or it helps in cognitive disorders. So this is the last slide I am going to share with you. So this is a small video which says in 60 seconds, 204 million emails are sent, 300K logins to Facebook, 1.3 million views to YouTube, 2 million Google searches, 1 lakh tweets, 20 hours of video, 62 hours of music downloads, happens just in 60 seconds. And when data has become big data, we talk about bytes, kilobytes to mega petabytes and petabytes, and the variety of the data is ever increasing, and the velocity and real-time streams are now extremely high, and it's the next wave of performance. And big data means a big opportunity for all of us, and the time to act is now. So with this, I would end my presentation and would like to take any questions if there is any.
Thank you very much, sir. You did a great presentation and very well voluminous and experienced presentation on big data biology in health and research with analytics. Thank you very much, sir. Now I request if there are any questions, audience can please type in the chat box or they can raise their virtual hand so that I can unmute them and they can directly speak to the sir and clear their doubts. If there are no questions, you can write to us later also or in future. If you have any doubts, you can mail us or write us or you can contact us so that we can revert them to sirs and get your clarification. Okay, sir. Is that okay, sir?
Yes, fine. Absolutely fine.
So let me check few questions in the YouTube, sir. If there are some questions,
Yeah.
I will ask.
Yeah.
So in YouTube also there are no questions, sir.
Okay.
So I would like to wind up today's session by giving a formal vote of thanks. First and foremost, I would like to thank you, Dr. Fakruddin sir and Dr. Suabrna sir, for accepting our invitation to deliver today's session. Dr. Fakruddin sir has given a wonderful session on the CRISPR, clustered regularly interspaced short palindromic repeats. Thank you very much, sir. On behalf of KSTA and on behalf of all the participants and on my own behalf, thank you very much once again. Also, I would like to thank Suabrna sir, who has given a very wonderful presentation on big data biology in health and research. On behalf of participants and on behalf of KSTA and on my own behalf, thank you very much, sir.
Thank you.
Amidst your busy schedule, indeed, it's a great pleasure to have you. You have accepted our invitation, sir. Thank you very much, sir. And also, I would like to thank our chairman, sir, Professor S. Ayappan, and our CEO, Dr. M. Ramesh, sir, and other senior officers and the technical team and non-technical team who are behind this workshop and the webinar series, lecture series. Thank you very much, sir. Now, last but not least, I would like to thank all the participants who have joined with us in this WebEx Cisco platform from various districts and colleges and universities and YouTube platform also. Thank you very much once again. You can join us tomorrow on by the session on vaccines by Dr. Shri Krishna Islur and a session on bioinformatics, Professor Kshitish K. Acharya. It will be starting at 3:30 afternoon and 4:30, followed by Shri Krishna Islur's session. Thank you, sir, once again. Thank you, one and all. I request you all to join tomorrow at 3:30. Thank you. Sir, if you are free, we can share you the link also, sir.